From Microbe TV, this is TWiEVO, This Week in Evolution, episode 46, recorded on August 30th, 2019. Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Hey, good to be with you from Elde Lab Studios, and back from our TWiEVO summer break.
Yeah, we did have a little break because we'd done extra work in San Francisco, right?
That's right. So the last time we were in the game was at the American Society for Microbiology meeting, ASM annual meeting, and we did two fun interviews back to back. Paul Turner and Talia Karazov. Those are now posted and running. And then in the meantime, we just kicked back on the beach and relaxed.
Yeah. Well, we didn't do any pods, but I don't know. I guess I can say I always relax, but I don't need to be on the beach to do it.
Oh, that's good. So yeah, actually it wasn't super relaxing. At least we crossed paths again in Minneapolis.
That's right. ASV.
At the ASV, where you're doing some video podcasting there. That was an interesting kind of historical look at the organization of the American Society for Virology.
Mm-hmm.
And then I heard rumors if my social media, sort of observations are correct, you've been doing some more podcasting in some exotic places recently.
Yeah, let's see. I was at Rutgers. That's pretty exotic.
Yeah, that counts.
And that one was posted. We had 4 guests on that one, and Dixon and Brienne came since we could all drive there, so that was good.
Cool.
Last week I went to Sweden. I did 3 TWiVs there, which will be coming out over the next few months. Yeah,
that one is the one I was thinking of. That looked pretty interesting. Some exploring in Stockholm, and where else were you?
So at Stockholm, I was at the Karolinska. They have a couple of different campuses, so I was on 2 of them. There's one in Huddinge, and there's one in Solna, I guess.
Mm-hmm.
I did a TWiV with 3 people at each institution, and then I went to a Swedish Society for Virology meeting in Smögen, which is on the west coast.
Beautiful. I'm getting jealous over here.
Little island right off the coast, really beautiful, and about 150 people, maybe 200. And I did a podcast with Erling Norby, who is a retired virologist, but for a number of years was on a committee that talked about who should get Nobel Prizes, right?
Mm-hmm, mm-hmm.
And so we talked about Nobel Prizes in virology, and that was really interesting. And he's actually visiting the U.S. in October, and we're going to do part 2 because we didn't have time to finish.
Oh, wow. Very cool. I can't wait. So that hasn't been released yet, but keep your eyes open on the TWiV channel. I think
that'll be released a week from this weekend.
Okay.
And he has a lot of insight into the process. And we started with the first Nobel for virology. Would you know what that is?
I don't. Yeah.
No, that would be for Wendell Stanley for crystallizing tobacco mosaic virus.
Really cool.
It was a chemistry prize.
Mm-hmm.
And we moved through some of the others, and he had a lot to say about all of them. 50 years after the prize is awarded, they open up the archive so you can actually see all the deliberations.
Huh, cool.
And sometimes people are nominated many, many times until they make it or not.
Yeah, right.
Some people don't make it, they just fade away.
Yep.
So that's fascinating. And so he's actually written a couple of books on this as well. So that'll be fun. And I'm looking forward to him coming back because we have a lot more to talk about. He said he's made many phone calls to winners.
Oh, he's the guy in the middle of the night when you...
Yeah, he's one of the guys who's done it, and he says you get a whole range of reactions. I would like to talk with him about that.
Yeah, great idea. I can't wait to listen to it. That sounds like a lot of fun. Did you also have a chance to do some sightseeing and sort of partying? They do these summer crayfish parties in Stockholm or in Sweden around this time of the year, mid-August,
so
late
it's
August.
Smögen, man. That's all we ate is is crayfish and shrimp.
Oh man, you're killing me over here, Vincent.
So they had these big platters of them, right?
Mm-hmm.
And so in Smiggen, we went one night to, they have all these boathouses on the water where you store your boat equipment, but they've converted them into cottages because people want to stay there.
Yeah.
And they let us go in one for a fest, big platter of shrimp and crayfish.
Mm-hmm.
And then the next night, Friday night, we went out on a boat into the archipelago, and they gave us the same thing, but we were cruising around. So that was a lot of fun.
Yeah.
And, you probably know this, but Swedes are nice. They're nice people. They're pretty friendly.
Yeah.
You don't get yelled at.
Yeah.
If you do something which in the U.S. would be frowned upon, they don't care. They don't say anything, So
Yeah,
I like it.
I love it. Kind of a shy, reserved people. I mean,
Yes,
your
yes.
your podcasting experience, it seems like a perfect match, a complementary match to sort of draw out these quiet Swedish people and get them to say something. Sometimes that can be a tall order to pull off. And so I'm even more interested to listen to those podcasts to hear some of these shy people actually go on the record.
Well, they're scientists. They're not shy.
That's good.
I think even as Swedish scientists, they have to be outgoing because that's what you have to do in science. Otherwise, it's hard to get places, right?
Yeah.
So they were all fine. They were all
That's
great,
good.
actually.
Well, and sometimes a shot of the aquavit can give you some liquid courage to speak up too.
Is that the Swedish drink, aquavit?
Yeah, it is. That's often so in these crayfish parties. There are some traditions where you'll eat a crayfish tail and then chase it with a shot of aquavit, which is sort of like it's sort of like a vodka-ish, but with some sort of different distilling traditions and herbs go in there. So that if you're in those crayfish tails, as you know, are pretty small. So if you do a dozen of those and you're actually following that up with a shot, that can go off the rails pretty quickly.
Well, we didn't have any of those.
Oh, okay.
I was asking about it, but they didn't have it. So we just had tails. Nels ate one.
Yeah.
It was, it's about 4 inches long and an inch in diameter. It was pretty substantial.
Okay. Yeah.
But it's too much work for me.
Yeah.
And all those shrimp, man, you need to
shrimp. Those will tide you over. At least there's some meat on the shrimp. The crayfish tails can be pretty modest, as you're saying. But we've got other invasive species, but we have tons of crayfish here in my backyard in Utah. So actually 2 weekends ago, I was out trapping crayfish. I got into about 200.
Wow.
Over a couple hours, and then boiled them up in the Swedish style. So you boil them in beer with a lot of dill, some salt and lemon, let it cool, and all of the juices kind of congeal together. And then you do the work of peeling those tails. It takes a long time. But I can eat, 30 or 40 in a sitting pretty easily if it's a nice evening.
So those are freshwater.
Yeah,
right?
fresh water.
The ones we had were saltwater.
Oh, okay. So these might have been the what are those? The langoustines.
I think that's what.
Yeah. Those are delicious too. Yep.
But I had a colleague at the meeting, Michael Lindberg, who's a virologist I know, and he showed me pictures of him catching crayfish at his summer house. Everybody has a summer house in Sweden.
That's right, and usually on the water, as you're saying. Yeah. Yep. Well, listen, I could talk about Swedish summer eating traditions and science all day, but we should probably get into our TWiEVO episode. So we have we're back after a couple of live interviews. We're back to hitting the literature. There have been so many cool papers just that have emerged this summer alone, some on bioRxiv. Really hard to just narrow to one today, but I think we've picked one that's pretty interesting. Resisting saying the word spicy that I've often
you got called out on Twitter.
Got called out on Twitter, but I stand by my description. Yep. And this qualifies. So we're looking at a paper. This emerged from. Is this in Science or Nature? This is in Science. Yep. Earlier in August. So the August 2nd issue. The title of the paper is "Somatic Evolution and Global Expansion of an Ancient Transmissible Cancer Lineage."
So Nels, who are the authors?
Yeah. Vincent, I thought you'd never ask. So it was a big team effort. Not, totally massive. Some of these certainly kind of the big genome scale analysis, which this counts in that category, can be really massive author list. This one's pretty big. It's led by a team, or the team, I should say, is led by Elizabeth Murchison. And there are 3 co-first authors, Adrian Baez Ortega, Kevin Gorey, and Andrea Strikova. And the whole crew is at the Transmissible Cancer Group. This is in the Department of Veterinary Medicine at the University of Cambridge in Cambridge, UK. I don't know how much longer we can say that it's the UK, actually. We'll see if that if that holds up in the coming months. But this, for TWiEVO, is a return to, I think, this really fascinating topic, rare topic, but still very fascinating and instructive, I think, in some of the evolutionary concepts of cancer evolution. And so we did a different transmissible cancer a few episodes back. I looked it up. This was TWiEVO 17, entitled The Curious Cases of Clam Cancers. Do you remember that one?
I do. My colleague, Steve Goff, right?
That's right. Next door to your recording studio headquarters for the TWiEV Empire. Were
you here at the time, or was just him that was in studio?
I'm remembering, I think that it was just him in studio. But I do know, at the time, the postdoc who was working on that project in Steve's lab, a guy called Michael Metzger, he's now running his own lab in Seattle. And it's a small research institute. I'm blanking on it. They change the name of it from time to time. I'll try to follow back up on that. But yeah, so he's taking these projects of these really interesting kind of lymph-related cancers that can be passed from clams, different species of clams, presumably just through they're kind of clustered together on the ocean floor or tide pool kind of scenarios, and the cells are transmitting between individuals in the population, and it spreads. It's an example of a transmissible or contagious cancer that moves between individuals in a population.
Yeah, you were here for that one.
Oh, I was here. Okay. Yeah. My memory is that's a couple years back. If we're on episode 46.
2017, March 2017.
Okay. Yeah, now it's coming back into focus. It was really fun to hear Steve talk about how that project emerged, and they really pushed on it in some cool ways. So, most cancer that we think about, and certainly is in the public mind constantly, given the prevalence of it in our own species, are non-transmissible. So the cancer emerges de novo, oftentimes in somatic tissues, so the skin or other, organ-related locations, and then develops. And there's a huge, basically more than cottage industry in cancer genomics. So taking tumor samples from folks, deep sequencing entire genomes or exomes. That's what we'll be considering with this transmissible cancers in dogs that we'll be talking about from this paper. But then, at the end of that individual's life, the tumor is gone. It as the patient passes away, so does the tumor. It doesn't have a source to continue on. And so in that sense, it's kind of, the evolution is really modest. It's only a several year process, and then it's over. The transmissible cancers, though, break that rule altogether. And that's what really, I think, caught a lot of attention on this paper. It's really fascinating. So, and the notion of a cancer being transmitted is the authors, I think, have a really nice analogy to the sort of process of metastasis that we think about in a single individual, where an emerging tumor or cancer cells will migrate to a different tissue, and then that can be sort of a bad prognosis. Here, the movement is not just within the tissues, but it's between individuals. And so this type of tumor or cancer is called CTVT. That stands for canine transmissible venereal tumor. And it's a malignant somatic clone that has been spread by direct contact and has been known for a long time. I think they note that it's in sort of, in our neighborhoods here in westernized countries, for example, it's pretty rare these days. And that's because dog populations are pretty well controlled. There aren't, feral dogs running around. And so just by sort of that kind of hygiene, you can really block the continuing transmission of these cancers. But over the last several hundred years, or as we'll see, even potentially thousands of years, this has been a kind of massively successful cancer in terms of its spread all around the world, and in fact, kind of circling the globe, as we'll discuss as they do some of the phylogenetic analysis.
Nels, you know that in New York City, there are tons of street dogs still?
I'm not surprised. I thought it was the rats that were the real game in NYC, but wasn't there a pizza rat from a couple of years ago?
So when I lived in the city, in the 80s, I guess, we were driving back from a restaurant, and it was like, I don't know, 2 a.m. We're driving through Harlem, and this pack of dogs was just running up and down the streets. So maybe like 50 dogs. Just wild feral dogs, So they're the ones that would spread these cancers amongst each other. I don't know if they exist anymore, but
yeah, good question. Would be more likely to. Actually, a great person to ask that question might be Colin Parrish at Cornell.
Yeah, it's true. Dog guy.
Yep, dog guy. He thinks more about viruses of dogs, so parvoviruses, et cetera. But he might have some ideas on that. So the other famous cases of this are Tasmanian devils. And that's been kind of splashed in the news over the last few years. And even in that case, because the Tasmanian devil population is so limited, possibly endangering the existence or sustainability of the species. That's one that was passed not through. So the dog venereal tumor is passed through sexual contact between dogs. The Tasmanian devil example is actually when they fight. So kind of face to face or nose to nose. But in each case, pretty extensive and dramatic contact, I guess you could say.
Yeah.
Right? Yep.
So there are two different Tasmanian devil tumors. Two different origins. There's one dog, started with one founder dog thousands of years ago.
Yeah.
And Goff told me this morning there are 5 known shellfish contagious cancers.
Oh, is that right? Wow.
And probably more, he said. Because,
sure.
That are what we found.
Yep.
And
known of so far. Yeah.
One wonders if there are human ones.
Right. Yeah. So far, I don't know of any strong evidence that way. And the authors point out that it's a fascinating case, but also the rarity of it. Although, how much have we really considered it? I think it'll be interesting to see if more cases emerge.
Yeah, for sure.
Yep. Okay. So, that kind of maybe sets the table a little bit. But so the real advance here, what the authors did, and again, a pretty massive effort in sort of genomic analysis, is they sequenced the exome. So that's the gene coding part of the genome, all of the, thousands of genes, a relatively small part of the genome actually. But so to save some genomic resources, they sequenced 546 of these exomes from CTVT the tumors collected from around the globe. And I think they say that the tumor samples were from something like 43 countries, which represent all of the inhabited continents. So I guess they probably don't have the Antarctica in there. There's a few dogs down there, I'm guessing, on some of the exploratory things, but maybe those aren't, constant residents, and it's still a relatively rare event, obviously. And so all these samples were collected between 2003 and 2016. So obviously a pretty narrow time window there. But using phylogenetics, and we'll get into the weeds on that in a few minutes, they're able to then try to use those modern samples that were pretty diverse to reconstruct what the ancestor might have looked like, even think about where it might have emerged based on what is known about dog genomics from normal cells. So they have a matching for this study in particular. They have a companion or matching set of 495 normal cell dog exomes that are tumor matched in the hosts. And from there, and then actually given some of the existing or other studies on even ancient dog remains that have been unearthed, we have a sense, or a growing sense, of what dog diversity looked like, not even from sort of the diversity of modern species, which is another fascinating area of sort of artificial selection, where we've done tons of dog breeding for traits that breeders and owners of dogs are sort of excited about. But there's these ancient samples dug up from Siberian permafrost and so forth, and so we do have there. There's some data there actually, kind of deep into the ancient genomics that is useful in this study. And so their goals are really to taking all of that existing, or that sampling of existing data, is to analyze the history, the spread, the diversity, and the mutational exposures that sort of collectively make up the evolution of this, what's inferred as a single original tumor clone.
And of course, because these are all one species, doesn't matter what dogs you have, right?
Yeah, that's right. And so you kind of have a nice, almost sort of context to it. You can really, it's really well defined, and because it appears, as a single clone, or sort of a monophyletic origin, it's you can start to apply some of the tools to try to decipher some of those characteristics, which would be probably more difficult, I would say, in the Clam case, or where you have 5 different origins. I mean, there you'd have to define that, or make sure you're counting for that, or the 2 origins of the Tasmanian devil case. It's still doable. It's just sort of a higher degree of difficulty. And then here, all the analysis can kind of go in the same direction under the assumption that this all came from a single ancestor. So, and already, just by before doing any evolutionary analysis, just by taking those normal exomes and comparing them to the samples that they've been analyzing, they note really a high burden of mutations. And so this is a mixture of SNVs, or single nucleotide variants. So this would be, the parlance is commonly the point mutation, where you just change a single nucleotide. A becomes a C, or a T becomes a G, etc. The whole kind of range of the 4 base pairs changing. And then they catalog a smaller collection of indels, or insertions and deletions. And in particular, I think they focus mainly just on small ones. So they aren't looking at massive kind of chromosomal or structure rearrangements. Those kind of things do exist quite commonly in cancers. It could be another maybe interesting additional or future work for the project. Are there sort of massive chromosomal changes that could crop up in any of these clones as they go on? I don't think they really address that in this one. But already, with just considering those 2 mutation types, they mapped something like 160,000 mutations. I think all but 12,000 of them are those single nucleotide variants, or SNVs. So then, it might be worth...
And these are mostly, I don't know if you said this, they're somatic mutations mainly, right? Correct.
Yeah. So, and that's a good point. So most of the mutations are on the sort of the autosomes, the 22, 23 chromosomes that are not the X or the Y. Actually, did you see in there? I didn't notice it. Whether does is the tumor from a male or a female dog? Does it actually say?
No, it doesn't say.
Yeah, I'm curious about it. I mean, I think you could diagnose that by the presence or absence of a Y chromosome, but I didn't dig down into that detail.
Yeah, because this should be independent of the host, right?
Correct. Yeah. So then, and I don't know a lot about the transmission, except that it is through sexual contact, but I believe it does pass, obviously, from male to female, and then female to male.
Yeah, they don't mention that at all. No.
But that's not included. But then, kind of putting it into the context of cancer biology, there's these sort of well-known concepts of the types of mutations that you might be interested in. So the main parlance is either a passenger mutation, which is something that's thought to be relatively neutral. It could just be, things are mutating all the time, especially in cancer scenarios. So if like DNA repair, is compromised in some way, the cells, the genomes will be accumulating, or they might accumulate the same number of mutations, but they may not be repairing them as well, for example. And so you'll have a higher mutational load. A smaller subset are the so-called driver mutations. And so that actually might be a mutation that disables some of the repair pathway, for example. And so that would be thought to be under potentially positive selection, or it would be beneficial to the tumor, sort of the selfish clone of genes that's out replicating the germline, in the sense that that is sort of the fuel, in this case, for more mutations to arise. You start to lose the growth control. And so these are considered the so-called driver mutations. And so that's an active and ongoing area of research in cancer biology, to understand what are, if you can sequence up and down the street on these tumors, but and as they identified in this set, just a ton of mutations. But what are the ones that you might really care about that are contributing to the growth control phenotypes, such that you might be able to modify or treat, that to block the ability of this to happen. So, and then those kind of ideas of passenger and driver then kind of fit somewhat cleanly into the evolutionary concepts, or positive and negative selection, or sort of neutral in between, in a sense. And so something that's positively selected, it would be this category of things that are beneficial in some way to the over-replication of the cancer or the tumor. Whereas something that's negatively selected, that would be something that's, or another term for this is purifying selection. So basically, in a mutational process, most mutations are deleterious. That's kind of almost cooked into our notion of what a mutation is, that it's bad somehow. That's not necessarily true, but a lot of them are. And so if those mutations show up and it's bad enough that you sort of break down the essential cell functions that are encoded by those genes that are mutated, those are quickly purified or removed from the population, or negatively selected. And so those are the main categories. The passengers, on the other hand, might be considered neutral. So you have a mutation that, so in this case, they're all looking at coding sequence, but it could be in non-coding sequence, which is the vast majority of the genome, or it could be in a coding region, but it could be a silent mutation that doesn't change the amino acid that encoded by the protein, and so encoded by the gene in the protein. And so then evolution or selection wouldn't really necessarily act very strongly on that. You can kind of get away with a mutation like that. And so that is also an interesting class. And a lot of evolutionary biologists, a lot of evolutionary theory is built on some of the ideas of what are, like, for the sort of collection of mutations, what are the primary forces in the evolutionary process. That's something that we've discussed a little bit. Matt Hahn came on the show. We had a fun interview with him. I forget the TWiEVO number, but it was episode we called Never Not Neutral, to try to be confusing about it, which I think represents the field as well, as they continue to argue or discuss the importance of thinking about neutral theory, for example. Anyway, so that's kind of sets the table, I would say, the kind of mutations and how that would fit into cancer biology. And then in this case, this really cool example where it's this transmissible cancer that goes back presumably thousands of years. But so that's what they really get onto next, is to use all of that comparative data to now take a phylogenetic approach to build the tree of the tumor based on the mutations or the changes they see. And from there, they can start to infer the ancestors. And so that gets us into figure 1. And actually, the figure takes up basically the whole page. And from the top to the right panel is just a massive tree of diversity from the samples. And then they're labeled by the location where the tumor sample is collected from. And then in the other panels, these really cool maps of the world, which they basically take the genetic differences or the collection of mutations, the comparative analysis, and then try to build a story about how CTVT sort of moved all around the world over the last several thousand years. So,
and you can do all that from contemporary samples, right?
Right, that's right. So in every case, they did note, I think in some of, well, and actually, we'll get to this point, some of the ancient samples, but I don't believe there have been any ancient samples. There's like one Siberian dog from thousands of years ago. No one has found an ancient sample that actually has this relatively rare tumor.
Mm-hmm.
So you kind of have to depend on all of those modern samples. And so you'd love to have that, that would be the best case scenario. And maybe, who knows, as, the permafrost is thawing out, maybe some ancient dog samples will be worth obviously keeping an eye out for that. Cause that'd be kind of the gold standard comparison that you'd love to make to complement a data set like this. But in the absence of that, you do the best you can to sort of infer backwards based on a lot of phylogenetic tools, including the mutational clock. So that's kind of one of the first things that they lay out in this study is that they propose that the tumor emerged, or the founder tumor might have originated from about 6,200 years ago. Plus or minus a couple thousand years on each side. So if you look at the confidence interval, it's pretty big. It's from 4,500 to, I think, 8,200 years. And actually it's a good, kind of cautionary tale or example of how difficult or tricky it is, or some of the limitations of just having these modern samples to really work backwards. So what they were doing is bringing together some fairly standard techniques. So they want to find a set of mutations, or between the samples that they think work on quote unquote kind of a clock-like mutational rate. And so these are things that you might expect to be relatively neutral. They're not getting selected on either strongly positively or negatively, which might then sort of be a faster or a different rate than just the kind of clock rate, which is the idea that any species has a mutation rate that just means a probability, or that much, 1 in 10 million times you'll have a point mutation, basically. So in this case, they pick just a very specific type of mutation. It's a C to T substitution at a so-called CPG site. And the motivation for that, or the rationale behind that, is that those are relatively well-behaved clock-like mutations that just crop up, and then selection, it's not always true. There are exceptions, but the assumption is that selection isn't having a major role in the retention or loss of those substitutions, those mutations. And then they use, there's people, and we've talked about this on TWiEVO as well, is the mutation rate, which you'd think would be sort of obvious, or like something you could solve for different species. It's still kind of, that's another active area of research, is figuring out mutation rates, even among our close relatives, the primates. Those numbers are constantly getting refined or revised as people do more sophisticated studies with genome scaled data. But they take the observations they make to the sort of one of the recent studies or assumptions on the dog mutation rate, and that's how they get to this confidence interval, or the idea that the founder tumor, in this case, showed up about 6,000 plus or minus years ago. And then from there, actually, it's kind of interesting. So there's this big sort of straight line where they don't infer a branch point for thousands of years. And so and that's based in part on the reconstructions and how the algorithms sort of return what they think is the most likely arrangement for how the modern tumors are related to each other, what the ancestors looked like as you sort of trace that back. And so, I think I take it with a little grain of salt, but it sort of is, at least intuitively, feels attractive in that you have this thing kind of smoldering in for thousands of years. And then it's only when, then it sort of takes off. And so they show the first sort of major branch around 2,000 years ago, again with 1,000 plus or minus 1,000 years, 1 to 3,000 interval on that. But then the real action that they're inferring is even more recent, something like 300 to 500 years ago. And now this might be starting to capture just how diversified the tumors from these dogs became as humans and dogs themselves started to travel around the world much more prolifically. So sort of the technology of maritime, as boats and ships were starting to move across the oceans. It looks like that's when this thing really started to diversify into separate populations all around the world.
Makes sense, right?
Yeah, definitely makes sense. You always, you want to be careful just not to follow too closely on the intuitive side, but that's what, I think that's what the data is saying. And then it kind of fits into the picture that you might expect. And so to really trace that origin, this is where that some of those samples, those ancient samples, came in handy. And so they suggest that in fact the original clone the ancestor of the tumor is a dog or a set of dogs that came from Central or Northern Asia. And that's because it just as a general match between that sample and dogs that have been sampled from around the world, it's actually has that sort of footprint or genetic signature of that genome. And from there, then they can start to say, and that's this sort of really cool, these maps where it has these arrows as it spreads, from Europe to North and South America. Looks like then they're inferring all these transfers back to Africa, independent transfers. So in the way that they build the case for some of the independent transfers, or sort of where it's not just sort of a simple, you just go to that geographic location once and spread out, is that the geography sort of breaks down in the arrangement of the tree. So for example, you see samples from India in one clade that's really distinct at the top of the tree. That's this yellow colored pattern. But then if you kind of go down the tree, you'll see interspersed in there from time to time other cases of Indian samples showing up that look a lot more closely related to ones in other locations. And that's how you infer this idea of sort of multiple or kind of independent or convergent appearances, is that even though the location is the same, the genetics is really different and actually looks like a different location. And that's sort of this sort of volatility of the transport of the tumor from dog to dog, as they were presumably kind of crossing the globe with their human owners. Yeah.
Yeah, it's amazing you can reconstruct this from these data.
I agree. Yeah, and
it looks just great because I would guess that people brought dogs on their boats when they started to travel, right?
Yeah, exactly. It's this sort of interesting, this crazy. We think about the impact we've had on the globe, and this is sort of an interesting, sort of surprising way that you can kind of see that through the indirect means, sort of through the dogs that we took with us that had tumors that were hitchhiking on the dogs that were transmitting through dog breeding, the dog breeding process. Yeah, kind of wild. So
you mentioned Colin Parrish earlier. He's interested in these canine parvoviruses. And it's a great story where years ago, a cat parvovirus changed by, 1 or 2 amino acids so that it could now infect dogs. And within a year, that virus had spread globally. Yeah. Through moving pets.
That's right.
Or as he said, stepping on poop in New York and being in Paris 8 hours later,
Yeah, that's right. Yep. Yeah, it's true. So I think it's a really cool example. Here, what's interesting about this case is a little different, right? So in this case, you can't just step on some dog poop and then, or have that interspecies transfer. So I don't think we know of any of that. This has really been restricted to, both the breeding process, but also somehow the way that those tumor cells then get established in a new host. Seems to be really host specific. And I think that's fascinating actually to think about, is that sort of those boundaries, how could it transmit from one host to another given diversity in the immune systems? But then that you have these species kind of these species barriers, at least as far as we know, and that this can't go to a cat, for example. It's really interesting. And then sort of a different level of kind of genetic exchange, or that the viruses are all over the place and have figured out ways to transmit cross species, cross kingdom in some case, and really prolifically even through the air on the mist, or those kind of things, versus in these cases of transmissible cancer, which maybe the shellfish are an exception, but certainly the Tasmanian devils. This is like life or death fights, basically, right? That are scratched up noses and so forth. So it's really interesting to kind of compare and contrast those systems of how populations can diversify.
Yeah,
given those biological mechanisms.
That, viral spread is just more efficient than a cell spread, right?
Oh yeah, definitely.
Because the dog tumors are not that common, and viral infections of dogs are way more widespread.
Yeah, agreed.
So having a cell established must be harder, right?
Yeah, that's right. And I think it's, reflected in the fact that we're just bombarded by viruses from all corners.
Yeah.
Whereas we only know of 8 cases of these kind of transmissible cancers
However,
to date.
Right? HIV is thought to be at least in part transmitted by cell-associated virus.
True. Yeah, true.
And it's very effective at transmitting. So something else is going on. I don't know if it's because you can also get cell-free virus transmitted with HIV or what.
Well, and I just think the efficiency of HIV's ability to package into those particles and then deploy in another cell, whether it's cell to cell transfer where you don't have to package and exit, or in cases that you do, that has evolved into such an efficient process where this sort of it's almost like a tumor transplant case, right? A higher degree of difficulty to pull that off somehow. Yeah. Okay, so then. That kind of sets the table just for the, it kind of defines the diversity of the samples and builds a really, interesting story for how this thing has moved around the world. Obviously, I think you're a little limited by the samples that you have, so there could be some just really consequential examples or mutational profiles that are just missing from your sample that could still emerge. I think that argues for maybe the story isn't completely complete, although to me, 450-ish samples sounds like a lot. And given that they've covered 43 countries, covered quite a bit of real estate, scientifically and literally. But then, now with this data set, they can kind of return and actually start to ask what are the mutational patterns that might emerge and might be associated with being, a tumor, something that's undergoing a ton of somatic mutation, and also how did it become a tumor? How does it persist? Are there some sort of genetic clues based on the types of mutations that are under the hood of all of these samples in the exome comparisons? So here they turn to this really growing and I think interesting data set that has just emerged maybe in the last decade or so in human cancer biology. This is the so-called COSMIC data set. I think there's a few other sort of major databases or repositories like this. This one, the COSMIC stands for the Catalog of Somatic Mutations in Cancer. And this is sort of contributing to, and I'm not really, no way an expert, but really interested in the little bit I've heard about this kind of some of these emerging ideas or the emerging field, is that you basically track patterns of mutations. So this is mostly coming from patients' tumor samples that have their genomes or exomes sequenced. And then using kind of sophisticated algorithms, you start to classify the tumors and looking for sort of shared patterns of mutation. And so there's some obvious ones, which are the oncogenes, which you'd expect, some sort of name brand driver mutations, so to speak, where these things that we show up again and again and contribute to the emergence of a tumor. But there are also just sort of main or kind of general patterns that can emerge that are put into these different classes. And this is having an increasingly big impact actually on cancer treatments. So traditionally, if you come into the clinic with, say, breast cancer or colon cancer, or pancreatic cancer, you'd be given a set of treatments that relate to cancers that have, that tissue type. You're treated by the tissue that your cancer emerged from. And so what this COSMIC data set and what others are doing now is saying, hold on, let's not worry about the tissue this came from. Let's worry about the pattern of mutations. And so if someone shows up in the clinic and they've got a lung cancer tumor, but it looks like a pancreatic tumor, and this is just a totally random example, but then you might treat that not on a lung cancer therapy sort of regimen, chemotherapy, radiation, et cetera. But you might use some of the drugs that have been useful or more efficacious in pancreatic cancers. You're basically treating the mutation type or the mutation signatures as opposed to the tissue. And at least for some patients, I think that's bent the curve a little bit on the effectiveness of their cancer treatment. Is as we get a better handle on the genetic similarities, getting a little bit closer to the mechanism of how these tumors are evolving or continuing to grow, or how they evade the treatments that we use against them, is turning out to be a really useful or increasingly useful approach to treating cancer. Here, I think the authors, nicely take advantage of that emerging data set to sort of put it into comparative context with these transmissible dog tumors. And so they actually identify a couple of signatures that are in the cosmic data set are close to that. And so this is in figure 2, and you can kind of see these peaks that show up for the collections of the types of substitution. So the C to T mutation that they note is part of one signature. Signatures 1, 5, and 7, I think, are the ones that their tumors match the most closely to the human data of tumors. There's something like 20 or more classifications or signature types. And so then people are starting to take those signatures and then trying to correlate that to the symptoms or the either the symptoms or the sort of sources of the mutation. And so some of these, I think it might be the signature 3, but don't hold me to that, is consistent with damage from UV light. And I thought that was really interesting. They also, another signature is consistent with a lot of ApoBec. So this is these sort of most familiar as virologists with ApoBecs as a restriction factor, but these are these cytosine deanimases that will actually mutate, in some cases, viral nucleic acids to try to as a way of as an immune defense, but can also be involved in mutating our own genes in our own RNA nucleic acid, depending on the situation. And so those are the two main signatures that they see. The other cool thing that they can do here, Vincent, is take advantage of that evolutionary tree again. And so they can actually look for signatures in the ancestors that they infer, and then compare that to the modern samples that they have already in hand. And so they note that when they did this, they found what's called signature A in one of these databases. And it doesn't go into a lot of details. And I, again, not an expert in this, but that signature has been associated with DNA repair shielding. So this might be, DNA mismatch repair, the whole suite of enzymes and complexes that are at work in our healthy, normal cells trying to counteract the mutational process so that your genome doesn't go off the rails, so that you don't end up with a aggressive tumor. And so that at least gives, and to say, so they see that signature in the inferred ancient samples, those thousands of years ago, that kind of smoldering CT-VT ancestor. But they don't see it in the modern samples. Those signatures are gone. And so they're using that as sort of this cool inference of an evolutionary process where perhaps that gives you some clues about how, as those cells that became this long-standing transmissible tumor, how they kind of came online as a tumor in the first place. That these are the driver mutations potentially, that you knock those out or mutate them in a way that then you lose your DNA repair shield. But then once the tumor is up and running and it's spreading all over the place and being passed from dog to dog, then you don't see that signature. There's that pattern of signature, of mutational signature. The process changes in a sense. And so the tumor, as it's gone on for thousands of years, has sort of switched in the kinds of mutations that are continuing to accumulate.
Didn't need it anymore, right?
That's right. Exactly. That's right. It kind of got established and was replicating, and then the types of patterns of mutations that are accruing more recently are different. It'd be I don't know how, if is that like flipping a switch or how subtle that is. It would be really interesting to know if that's sort of how sharp that is. And I just don't know enough about these signatures and how well you can trace them or put them into categories to say signature A versus not, or how much of a continuum that is. But that would be a cool, interesting future question, I think.
They suggest that maybe it disappeared as a result of a metabolic change in the cell. And one of their ideas is that in parallel, in other branches, they see a change in mitochondrial DNA, and that may be associated with that metabolic change that they're postulating.
Yeah, right.
This is also speculation, but that's an interesting thing to think about.
Oh, no, I totally agree. They also threw in a sentence or two that was pretty provocative, but I didn't see that they follow up. They said that they, related to your point on the metabolic change, but they infer the independent replacement. So in two branches, the replacement of mitochondrial DNA itself by horizontal transfer. Yeah. I thought I'd love to hear more about that, actually, or how they dig into that idea.
So how do they see a sequence or a signature that was present up to 2,000 years ago and then goes away because we don't have any 2,000-year-old samples?
Correct. So what they're doing here is they're trying to, good question. So I think what they're doing here is actually, it's a couple of comparisons. So you can compare quote unquote normal dog sample and the collection of mutations relative to you could take the modern dog samples, compare that to modern tumor samples. And then in parallel, you can do phylogenetics both in the tumor samples to infer what the ancestor looked like, and also in the normal samples to infer what the dog ancestor looked like. And in fact, and I don't know if they do it for this part of it, but the ancient DNA can potentially come into play here as a normal comparison point as well. Yeah. And so then you have basically a set of mutations both in a modern comparison and then in an ancient comparison. And from there, it's sort of two different comparisons. And then you ask, what are the signatures or patterns of mutations that are in both? And so that's in the ancient one, they saw that signature A, and in the modern ones, they don't.
Okay. So it does depend on having the ancient samples.
Or you could use the modern normals and then infer an ancestor using those as well. Okay. So I think either and to be honest, I don't know how they did it, but that would be my best guess. They do. I mean, and I think it's important actually to really think about this carefully, is especially when you're inferring mutations, and because that process, it's it's not, you don't have a time machine here, and so you're using algorithms, you're using statistical arguments. And so, if it's a kind of a big pattern across genes, then maybe there's something to it. But signal to noise, I think you always want to think that through. And in fact, they have some nice signals that they highlight that I think also kind of adds to the story, both in interesting ways, but also maybe gives the data some robustness, or gives it a good sort of reality check in some ways. And so one of these gets back to some of the signatures that are often sort of crop up as a reaction to UV damage.
Mm-hmm. It's very cool.
I agree. So they have this really interesting correlation between, because they have hundreds of these samples, as we said, from all over the world. And so they looked at the geography of it. So how close are you? What's the latitude? How close are you to the equator? And whether or not there's a correlation to the patterns of UV damage, basically, or that you commonly see in a lot of tumors. And in fact, they do see a pattern correlation. So the stronger patterns of UV mutations in the samples were from ones that were closer to the equator. And they note that in the just sort of life cycle in a way of the tumors, that in fact the way the tumors grow or are deposited, that they actually can be exposed directly to sunlight in some cases. And so I thought that was a really fascinating sort of facet of the paper.
Yeah, it's cool. I like that. Estimating the latitude of the founder. Yep.
Yeah, exactly. That's pretty interesting. Okay, so then finally, this kind of gets at the types of mutations that have been sort of driving clinical practice in new and exciting ways. But then to think, again, sort of in a little bit more of an evolutionary context, what kind of selection has been acting potentially on the mutations over these thousands of years? And so, and this is a case where they point out that the sort of cancer biology as usual sort of goes out the window in a sense, because as we were discussing a little bit earlier, most of these, most tumors don't transmit. They show up, they quote unquote evolve for several years, or the lifetime of the individual that has the host of that tumor, and then go away. And then that process plays out again and again. And so from an evolutionary standpoint, that's actually, that's quite a bit of a challenge, because you have all these independent observations, but you don't have a lot of time to see how evolution might sort of select, how natural selection might unfold on tumor biology. And so I think that's where this data set has, whether it applies directly to thinking about diversity of human tumors, I don't know, but it does give you a new window, in a sense, to look at how natural selection might act on tumors over thousands of years, since this is kind of a long-standing clone that is diversified all over the world. And so they first, can see, again, getting back to sort of that driver idea, some of the brand name oncogenes, the MICs, the P10s, the RBs, the so forth, and it's kind of alphabet soup. But these are well-known cancer genes that can have these so-called driver mutations. You basically can have a direct biological link to a lack of growth control from mutations. Those are still in evidence in the modern samples and inferred all the way back to some of the ancient sequences of those genes that they put together. They also then take kind of a common set of tools commonly used to look at natural selection. This is the DN-DS ratio. We've talked about this in some past episodes. This is mapping sort of the outcomes of mutations, the types or rates of mutations to infer something about natural selection. So this is the rate. The DN is the rate of the non-synonymous change. So that's a point mutation that leads to an amino acid change versus the rate of the synonymous or silent changes, the DS. And so this is an outcome of the degeneracy of the genetic code, that you can have some point mutations that you end up with the codon that encodes the same amino acid, even though it's a different codon. And so just by taking that ratio, you can start to make inferences about the selection that might be acting here. And so if DNDS is high, meaning you have an overabundance of non-synonymous changes, that's indicative often of positive selection, where there's some beneficial outcome. In this case, it would be for the tumor, in some way, the fitness of the tumor, so that it continues to propagate or to do well. It's a beneficial mutation. Versus a low DNDS ratio, now you have very few substitutions that change amino acids. And the notion here would be that it's not that those mutations didn't happen, the non-synonymous. It's just that if they did, they were so bad for the gene that encoded the function, or that ultimately allowed the tumor to continue to live or divide, that those are culled out of the population. If you have an intermediate level between the two, sort of a DNDS ratio around 1, that can be a neutral scenario. So you don't select the mutation in or out of the population. And in fact, that's probably what they're seeing here, is mostly a combination. So very little positive selection in the modern samples. And the inference is early in the life of a tumor that is the driving force, that you have these driver mutations that you select on very strongly, because this is how these things over-replicate or break growth control or evade the immune system. Here, it looks like they propose that in their samples, the modern samples of this, thousands of years old tumor, that in fact, most of the DNDS values are intermediate in the genes. And so they propose that you don't have a lot of selection acting currently, and surprisingly not a lot of negative selection either, meaning that the fitness might be on the decline is one of the outcomes. And maybe, as this clone has gone for thousands of years, it's not getting stronger and stronger, and it might not even be sort of staying at the same evolutionary fitness, but it might just sort of peter out, might give out, is the inference that they're drawing, or the speculation that they're making based on the types of mutations that are accruing overall. I'd say this part of it, to me at least, was maybe the hard to draw any really sharp conclusions. There's also some limitations of the DNDS as a tool to think about natural selection. It's just considering point mutations. Again, the indels, the massive copy number variation or genome rearrangements, could have a lot to say about how natural selection is acting, and they don't even, it's much harder to do that analysis, but that's not part of not considered in this study. But I think maybe the strongest conclusion that they can draw is that they don't see obvious signs of ongoing strong positive selection, meaning that, the tumor sort of potentially reached this plateau of existence, and the transmission sort of works, and that if there's not a lot more sort of selective headroom one way or another for the tumor to continue to expand, or for the fitness to be gained, for it to become more aggressive, or to find more beneficial mutations.
Now, this idea that it's the cells are accumulating mutations, and fitness is declining. Is that because there's no horizontal gene transfer that would rescue any deleterious mutation?
Yeah, so that could be, that's certainly the case, or some of the ideas or theory around the idea of asexual reproduction.
Yeah.
You're exactly right, Vincent. And so, and I think that, that fits into that exact idea, which is basically, why go through reproduction? You're losing half of your genetic contribution in the next generation.
Yeah.
And yet we do it. And almost everything does it. The exceptions to that, the idea is that they're in sort of potentially a dead end. And because you're not, just like you're saying, recombination can be a powerful outcome of sexual reproduction, which then sort of can relieve the mutational burden or can make new combinations of alleles that can be selected on as well in new ways. Mm-hmm. And so I guess maybe the irony for these venereally transmissible tumors is that they depend intimately in sexual reproduction, but they don't take advantage of that genetically. They just pass asexually as a clone from generation to generation in a sense. Yeah.
That's a good point. Yeah.
Yep.
Yeah. That's, last time we did the Clam tumors with Goff, we referenced a paper or an opinion he wrote for PLoS, which was basically these cancers, these transmissible cancers are like a sixth type of pathogen.
Yeah.
Right. Mm-hmm. Let's see if I can figure all five out. We got viruses. We have bacteria. We have fungi. Mm-hmm. We have protozoan parasites. Mm-hmm. And then the tumors would be the sixth. So what's the fifth?
Is it prions maybe? Is it infectious protein particles? Or is that, I guess
proteins. I guess that would be it, yeah. Okay. But I should check.
Not technically alive, although neither are viruses, depending on if you want to delve into that controversy. But
well, he calls it a sixth modality of infectious disease.
Yeah. So I think I would put infectious proteins in there, prions.
That's a modality of infectious. Now let's see if we're right. So we got viruses, bacteria, fungi, parasites, and prions.
All right.
Yeah.
Beautiful.
Good job, Nels.
Pass the pop quiz.
Yeah, it's a good question for an exam. What's the sixth type of
I love it. Yep.
Organism. I like that very much.
Yep. Bonus points.
Now, this statement they make, transmission bottlenecks between hosts render weak selection inefficient, which then they say leads to accumulation of deleterious mutations. Why doesn't something emerge that overcomes these bottlenecks? In other words, you could make the cells more transmissible, right?
Yeah, right. And I agree. And so that's, so far for whatever, and part of it might just be mechanics of how the process started, right? And so then how do you go from, depending on sexual contact to be transmitted, which is a pretty specific type of interaction, to something that's more promiscuous? That could be a pretty strong barrier that at least, and again, if we put this into some evolutionary context, 8,000 years on the sort of high side of their estimate is still not very long. And so,
that's true.
Yep. And so
for a virus, a transmission bottleneck could be overcome by making the particles more stable or by just making more of them, right?
Agree.
And there are other ways too, but those are two. And so for these cells, maybe it's telling us that, it's not easy to make more because you can only divide once every 24 hours, right? Mm-hmm. So you can't make billions and billions of particles. And maybe it tells us that stability is not an issue in transmission, right? Because it's venereal, so, you don't have to be exposed to the external environment, so you don't have to get more stable.
Mm-hmm. Although, it is interesting that UV signature, right? So that there is some impact of environmental exposure that they're...
Yeah. They say that the tumors usually occur within the genital tract, but sometimes protrudes. Mm-hmm. Okay, so that would give you exposure to sun.
Yep, exactly.
Yeah.
And that's kind of another cool point that I think they draw is how basically by collecting... It's almost like I think they describe it as a biomarker of the environments that these tumors have seen over thousands of years based on,
that's right.
based on the kind of environmental exposures, including to the UV. Yeah.
So I think it tells us that, these cells go directly from dog to dog. So, they're not floating around at all like the Clam tumors or viruses. So there's no advantage to becoming more stable. They're already there.
Mm-hmm.
And they say that in the discussion, they may have optimized their transmission early in history, and that's it.
Yeah.
They can't get any better.
Yep. Yeah, it's hard to say. I mean, the other thing is, well, in this case, I mean, honestly, the sort of behavioral modifications, right? If you just, and this is Mark's human intervention, but if those streets of Harlem are not running with 50 wild dogs, or the story you were describing earlier, then it sounds like I think the transmission goes way down. So that's the observation, that it's really hard to get modern samples, or increasingly difficult, because as you have, the more dog catchers you have, basically. And so as, I guess as dogs are, as a population are more pets than they are running wild, then the natural ecology or biology of those interactions goes down, and that might actually have a really strong impact. So if I were, a really kind of aggressive CT-VT clone, I'd figure out how to convince the dog's owner to allow the dog to have as much sexual contact with other dogs as possible.
That's it, because that tells us why there's only one lineage also, because the contact is decreasing. Whereas with clams, there are 5 and probably way more, because there are billions of clams, right? I don't know if there are billions, but maybe hundreds of millions.
Oh, yeah. Yeah. Yep.
I mean, they do live in more shallow waters, right? They can't go way down.
Correct. Yep. Yeah. Interesting.
Yeah, it's very interesting.
Yeah, it was fun. Yeah. Agree.
So the thing, once this goes, then probably that's it, because the contact is between dogs is declining.
Mm-hmm.
Right.
Yeah, and maybe that's reflected in some ways in some of the mutational signatures. Although I'd be a little cautious to draw too sharp of a line between those observations at this point.
One thing that I think is worth pointing out, that this signature A, right, that the one that was there and then went away, they say around the same time there's a signature in the human genome. It's in the germline, actually, which came and went also. And they're wondering if it's a shared environmental exposure that was there and now it's gone.
Oh, interesting.
Humans and dogs probably shared environments for many years, as they do now. Right?
Oh, yeah. And especially in the last 10,000 years or so. I'm forgetting the exact date of domestication that's estimated. But yeah, no, that has been an unusual sort of coevolution in the domestication of dogs. Our two species are really tightly linked. As we've become really sort of, enamored as pet owners.
So the dogs and wolves diverged 20 to 40,000 years ago.
Yep. Yeah, well within the time. So that well within the time of the emergence of this tumor.
Yeah, for sure.
Mm-hmm.
You wonder if it was us that really drove this emergence, right?
Yeah, well, certainly. I think the type of interactions between packs of dogs from the sort of non-domesticated wolf, ancient wolf era, the density of, as soon as you start domesticating and breeding, population density goes up, the amount of exposures, the artificial selection on controlled matings. I mean, that goes through the roof. And so, yeah, I think that's a real part of the equation there.
I mean, in addition to us transporting them, right?
Oh, absolutely. Yeah. And that's really with all the diversity they sample, I mean, that's the real game here, is the distribution of the tumor around the world as a hitchhiker on dogs as companions of humans that have basically stretched out and intermingled for thousands of years. Or actually even the last 500 years, with a really dramatic increase in trans-world travel.
So this brings the question of whether, back to something we brought up in the beginning, are there human transmissible tumors? And, you just need to look at some and you'll get the genetic signature, which says, oh, look, this is clonal. But I don't know if there need to be, because we have viruses that, like HPVs, right? Sexually transmitted tumors.
Correct.
So I don't know if you need, if a cellular transmitted tumor would survive in that niche, I just don't know.
Yeah, I don't know either. I think that pulling off that transmission sort of through a behavioral exposure, obviously reproduction is one that will, that our species will continue doing. And that's, in that sense, so in contrast to the Tasmanian devils, like we're not, as part of our behavior, it's pretty rare, actually.
We don't bite each other, right?
Bite each other in the face, thankfully.
Yeah. So sexual transmission would make sense for us, but also maybe respiratory, but it would have to be close contact, because a cell is not going to go too far, right? In an aerosol, I would guess.
Yeah, agreed.
It's going to weight it down. So maybe some lung cancers are transmitted among families who are close together. I don't know. It's an interesting question.
It would be interesting to look at. I think here, obviously the immune barriers come up. And so that'd be another angle that I think would be really, and maybe there's some, details hiding in the dog data, is how do you, so that the notion of, a graft rejection, I think, comes into play.
Mm-hmm.
And so how is it that these cells aren't recognized by the immune system as, especially as it diversifies, as something foreign? Like, is there selection on that so that it remains like this? It's basically just your own cell.
Mm-hmm.
As the host diversifies and the tumor diversifies, you could imagine that then an incompatibility would sort of, or sets of incompatibilities could emerge where the immune system says, hey, this isn't me, and let's get rid of this thing. And so that could be part of the barrier, I think, in these transmissible cancers. Whereas the sexually transmitted viruses, et cetera, those ones, just as part of the replication program, you're selecting in really strong ways for changes in growth control that can move into oncogenesis as well recognized. Seems like certainly a more successful path. But yeah, whether both exist, I think it's a fascinating question.
Yeah, people are looking, I'm sure.
Oh yeah. Yeah. Maybe this will be TWiEVO episode 460.
It might not take that long, It's a long time.
That's a long time. I don't think
many years. We do.
I don't think we'll be podcasting.
We do 50 a year, roughly. No, not 50.
No, 12.
We do 12.
Yeah, so
12 a year. Yeah. Well, you're going to keep doing this, right? That's what you told me.
I am. I'm all in, but my days are numbered too. So
maybe mine are more numbered than yours. That's for sure. Well, get yourself another co-host. If I go,
we need to.
Whenever I go, you're going to figure out how to record. Well, you can probably do that.
Yeah,
I should teach you how to edit.
Absolutely. Yeah, we'll keep it. Next time I'm at headquarters at recording studios, we can start to do that, and hopefully, there's a young aspiring podcaster out there who's already thinking about stepping into our shoes at some point. But we'll see if TWiEVO is transmissible or not.
I'm sorry, I should have thought of this, but I'm going to be in Nevada in September, but I should have stopped by in Salt Lake. Yeah. Because I'm flying through somewhere. I don't know where I'm flying through.
Well, let me know. We could grab dinner or something if it's through Salt Lake. And then we're going to, as a preview, we're going to be together at a meeting I think it's in November.
That's right.
Near Munich, the giant virus meeting.
Mm-hmm.
And we'll be doing some podcasting from there. I'm really excited for that one.
Yeah, that's a very interesting venue. You'll like that. I've been to that once before.
Mm-hmm.
And the meeting is full of very interesting people, so that should be a good episode. And Rich Condit will be there, right?
That'll be really fun. And just that field of giant virology, I think, is really an interesting one from both strict virology perspective, but also from a strict evolutionary perspective, kind of all of the above. Yeah. They're really interesting. Yep.
Cool. There are a couple of emails. Let's do them.
Oh, great. Yeah, let's go to the mailbag here. So I can do the first one. Irene writes, Dear Vincent and Nels, greetings from Durham, North Carolina, where it is currently 68 Fahrenheit. That's 20 degrees on the centigrade scale, and sunny this morning. I just wanted to thank you for featuring CRISPR and ants. That was TWiEVO episode 18, 2 years ago. I just led an undergraduate seminar course on EVO-DEVO, and one of the topics we covered was centered around using CRISPR to understand evolution. The students chose to read this paper. I gave them 3 papers to choose from, and they really enjoyed reading and discussing the paper. This was, and just to say I'm interrupted, this is from Dan Cronauer's lab at Rockefeller. The episode is the Raiders of the Lost Orco. I think maybe the best TWiEVO title to date. Using CRISPR to disrupt this gene Orco. Involved in the behavioral process. So it was nice to finally read the paper and revisit the podcast episode. It was also a nice paper to introduce aspects of neurobiology and neurobehavior in our course. We hadn't read any papers in that area, so it added to the diversity of topics that fall under the umbrella of Evo Devo. My pick, in honor of, so we have a listener pick here. I'll go right into it. In honor of Fascination of Plants Day, which will happen tomorrow. This is a couple months ago. We're a little behind in the mailbag at the moment. Thank you for all of the letters, and we love to get to them when we can. I would like to direct people to @EndlessForms on Twitter. Dan Chitwood and colleagues post amazing CT scans of plants and more. For more plant coverage on May 18th, which was the Fascination of Plants Day this past year, you can find tweets under the hashtags, and that's hashtag Plant Science Fascinates Me or hashtag Plants Day. Many thanks for bringing to attention cool studies in evolution. Best, Irene. Thanks a lot, Irene, for your note. And I'm thrilled that the Raiders of the Lost Orco episode on ant CRISPR techniques, this pioneering work out of the Cronauer lab, caught the attention of your students there. That's really exciting. Also, I'll definitely check out this @EndlessForms. And as we've increasingly recognized and strive to do, the evolution of plants is absolutely a massive part of the cool biology that we're excited to share. And so thank you for your listener pick as well.
I'm sorry, Irene. I don't know why this escaped me since May, but my apologies. And I think Irene had written about us doing a TWiEVO at some evolution meeting, right?
Yeah, that's right. I'm trying to remember if that could have been SMBE, which is the sort of annual big molecular evolution meeting. It was this last summer. It was in Manchester. I think it overlapped with ASV.
Okay.
But then there's also a major evolution meeting. I think it was on the East Coast this year that I didn't make it to, but it would be fun in the future, just as some of the TWiV Empire podcasts show up at meetings. I would love for us to expand into some of the evolutionary venues as well. And
yeah. So if you want to try again, Irene, just let us know. Maybe we should work a little sooner, get in touch with the meeting organizers and so forth.
Yeah.
I don't know. Is it always overseas or?
No. So SMBE is it's kind of 50-50 international and then in the US. And then I think same for evolution, the kind of major meeting that's not just molecular evolution, but it also kind of covers all the way to ecology and evolution as well.
Okay.
But either would be great possibilities, or other meetings that are evolution focused. I think as people either reach out or also I'll start to try to get that on my radar. I agree. If we should reach out early and often and see if we get any interest. It would be really fun to bring TWiEVO to some of the evolution meetings as well.
Right. John writes, "Hi, Nels and Vincent. My name is John. I'm currently a research assistant working at Oregon National Primate Research Center. Just want to extend some appreciation for the show. While I don't currently work in the field of evolution, I have a passion for molecular evolution, and your show has continually fueled it over the last few years. Keep up the good work." Thanks, John.
Yeah, thanks, John.
Glad you like it.
Me too. Oregon National Primate Research Center. I would, put it out there that primate diversity, even if you're not formally thinking about evolutionary ideas, it's kind of all around you. And primates in particular, in a lot of cases, vanishing diversity, where a lot of endangered species and we know very little about a lot of this biology, the diversity, even in our closest relatives. And so it's a really important topic. Okay, next one. Anthony writes, congratulations for being an inventor of Arabidopsis, which is not a weed. So to explain Anthony's point, he also appends some work from the poet Ralph Waldo Emerson, from his complete works published 1904. This is volume 11 under miscellanies, section 30, The Fortune of the Republic. I'll read this paragraph. It won't be as poetic as Emerson, but I'll do my best. And what is cotton? One plant out of some 200,000 known to the botanist, vastly the larger part of which are reckoned weeds. What is a weed? A plant whose virtues have not yet been discovered. Every one of the 200,000 probably yet to be of utility in the arts. As Bacchus on the vine, Ceres of the wheat, as Ascarite and Whitney are the demigods of cotton, so prolific time will yet bring an inventor to every plant. There is not a property in nature, but a mind is born to seek and find it. For it is not the plants or the animals, innumerable as they are, nor the whole magazine of material nature that can give the sum of power, but the infinite applicability of these things in the hands of thinking man, every new application being equivalent to a new material.
So this comes because of our conversation with Talia Karasov, right?
That's right.
Arabidopsis. I asked her, isn't it a weed?
That's right. So Talia would be the inventor of Arabidopsis, I think, right, in this poetic construction.
So while I was at Rutgers a couple of weeks ago for the TWiEVO, I was in the library, a little departmental library, having lunch, and on the shelf were two journals, scientific journals. One was called Weed, and the other, I think, either Journal of Weeds or Weed Journal. Two different journals devoted to weeds, and I always thought weed was like a derogatory,
right,
non-scientific, but obviously plant biologists call them weeds too.
True.
Isn't that funny?
Yeah. We need to broaden out that definition a little bit. That's great.
Okay, now it's time for some picks.
Yes, so we have our science picks of the week. So my pick is hearkening back to our opening conversation about your trip to Stockholm. So as I was on the social media platforms, I noticed some exciting photos that you had posted at the Vasa Museum in Stockholm. So that is my pick of the week, is the Vasa Museum. You were just there, it looks like. It's one of my favorite museums. So the Vasa is an old Swedish ship. I think this is the early 1600s. I should let you explain, since you were just
go ahead.
You were just there.
1628.
1628. So it was this massive warship that the Swedish king had commissioned and put together. They built it. Beautiful ship, sort of art, technology, science, warfare all together. Wooden craft, all kinds of heavy cannons and ornate decorations. So on its maiden voyage in Stockholm Harbor, the Vasa started sailing, and a breath of wind came up that tipped it over and sunk it on its opening excursion. And so, first of all, I have to say I just love that story, just from a sort of, as soon as Swedish people get sort of, really confident and flamboyant, you just sort of fall on your face and your big investment goes out. It's a great lesson in humility somehow, I think, coming from the Swedish background. But then the ship sank and sat for hundreds of years in the bottom of Stockholm Harbor, but was somehow preserved. So it avoided, aren't there like shipworms or something? They're supposed to eat all that wood, but somehow the composition or the mud it was in, it was safe.
So I asked the guy that we took a tour.
Yeah, awesome.
So this came up because for some reason Dixon on TWiEVO a few weeks ago said, or months ago, he said, the Vasa sank in fresh water. That's why it didn't rot.
Mm-mm.
And Kathy Spindler said, no, it was salt water.
Mm-hmm.
And the guy at the museum said, no, it was brackish water, which means it's a mixture of both, but there are no worms in this area, so that's why it was preserved.
Yeah. So the Swedish people hundreds of years later, sort of celebrated this failure by reclaiming, bringing the Vasa up to the surface from the mud and then restoring it and then putting it in this museum. And I think it was in a temporary museum for many years, and now it's in its more permanent space. It's a really great museum. Just
wonderful.
Yeah,
it's
really interesting. I've been there a few times, and every time I'm in that part of the world, I try to make it because it's, I don't know, I just really am inspired by it. But it's also, the story's not over. So the ship is in relatively great shape, but not stable. And so, in some of the ways it was originally preserved were not sustainable. And it is like slowly crumbling sort of right before your eyes in some ways. And so I think that's also an interesting ongoing story, somehow as well. So
yeah, it's so interesting because they had so it was saturated with water, of course. They had to replace it. So they replaced it with polyethylene glycol.
Yeah.
Which they just basically sprayed it with that for 2 years.
Mm-hmm. That's right. Yeah.
I guess that wasn't optimal.
Yeah. Now you've sort of solved one problem, but created a new one.
So there are a lot of interesting things about this, but what I found curious, so a number of people died on the ship.
Mm-hmm.
They were below decks and they were firing the cannons. And that's why it sunk, actually, because the cannon doors were open.
Yeah. And
otherwise it would have sunk out at sea, probably.
Yeah, right. It just would have delayed the inevitable.
So they got trapped under the cannons. They were on the lower decks, and they were able to retrieve a number of those skeletons, and you can see them there. But they got teeth, and they got brain tissue as well. And that's somewhere preserved, and people are doing some genomic and metagenomic, metagenome, microbiome analyses on them, which could be interesting.
Yeah, or finding if there are any microbes that are genomes that are still in there. So yeah, thank you. You kind of rescued this as a science pick. Kind of.
Well, they have their teeth, so they could go in the pulp and look for any bacteria that were coursing in their blood.
Yeah.
It's a common thing to do.
Mm-hmm.
But the brain tissue was intact, they said. So they would give it to Svante Pabo, who, of course, is a Swede himself.
Yeah, now we're talking. That's right. Right in his backyard.
And he could look at it, yeah.
Yep.
That's great. So I saw you had put a Stockholm pick, so then I put my pick, which is the Nobel Prize Museum in Stockholm.
Oh, yeah.
Have you ever been there, Nels?
Yeah, I was there once, a couple of years ago. It was funny because it was almost like visiting my neighbor one floor away from me, Mario Capecchi, who won 1/3 of the Nobel Prize in, I think, 2008. So I could go to Stockholm to visit Mario, whose lab is just a floor below mine here in Utah. But yeah, that's another interesting one. So that's in the Old Town area of Stockholm.
Yeah, it's beautiful.
Yeah, I do too. So you've got all those kind of cobblestone roads. The shops are all kind of packed together, and you're just walking through these alleyways. And then one of the courtyards, I think it's just across from a church, is it's in a lovely old courtyard lined with cafes, and then there are some old buildings around it. That whole old town is great. Yeah.
So the Nobel Museum is nice. It's rather small, but you can see some artifacts and stories and so forth. It's actually going to move. My Erling Norby told me they're looking for a place to move.
Oh, is that right?
In the next few years, so it's going to get a little bigger.
Huh.
But for example, had Liz Blackburn's dress that she wore at the banquet.
Yeah, that's cool.
He donated it to them.
That's neat.
And they had Ralph Steinman's tissue culture microscope.
Wow.
And on it, there were 3 buttons, the kind you pin onto your shirt, with dendritic cells on them.
Oh, cool.
So there are lots of cool things.
Yeah.
There. I mean, I because I was hanging around with Erling, I heard a lot about the Nobel Prizes, and it's very interesting. Hopefully it'll all come out in our series of TWiEVs. It's a very interesting thing. And
yeah,
course,
I can't.
They start talking about it in January.
Right.
Nominations start coming in. They talk it throughout the whole year. And he said sometimes they don't make the final decision until the morning of the phone call. Can you imagine?
Yeah, it's exciting. Yep.
And they used to, of course, send telegrams. They have a few of the old telegrams there, but now they call people. So it's a cool museum if you're in Stockholm.
Very cool. And a great cafe, if I'm remembering right, in the basement of it, you can get, one of these impossibly simple but delicious Swedish open-faced sandwiches for lunch or whatever. It's
And of course, if
great.
you're in Stockholm and you want to do the Nobel thing, there are two other things. One is if you go to City Hall, there is a room where they have the banquet of the Nobel Prize, and that's very impressive. And you can get a guided tour, and they'll talk about it. And then there's a small building on the Karolinska campus, which is called the Nobel Forum, and that's where they announce the prizes. They have a press conference there in a room full of journalists, and people get up and announce them. And you can see that live streamed now, actually. But it was cool to be in front of that.
Yeah, very neat. And again, looking forward to those TWiEVO episodes. Stay tuned. It should probably be a week or so, I think, when this releases.
So the first one with Erling will release a week from, today is August 30th. It'll be a week from this Sunday, which is September 1st. So that would be September 8th, right?
Yeah, that's right.
If I do the math correctly.
I think your math is good, and I will tune in myself. I can't wait to hear that.
All right, that is TWiEVO 46. You can find it wherever fine podcasts or not-so-fine podcasts are distributed. And if you listen on your phone or tablet, please subscribe. You get every episode, and we know how many people are listening, which can help us to raise money. If you want to send us a question or comment, we'd love to hear it. TWiEVO@microbe.tv. And please consider supporting us financially. Microbe.tv/contribute. That helps pay our expenses, and we like to travel. And as Nels and I were saying, we'd love to be able to go to some meetings.
Yeah.
And often we can get support for one person, but not both of us. So
true.
I can't do a TWiEVO on my own. Nels could, but it's more fun together.
It's more fun as a team, as we saw in San Francisco earlier this summer. And we really appreciate, it was fun. And we really appreciate all the support we already have and the kind of growing listener base, but we would love it if you could pitch in and join us on this science adventure.
You can find Nels Elde at Selvolution.org, and on Twitter, he's L Earlybird. Thanks, Nels.
Thank you, Vincent. Good to be back off of summer break. We're into the fall academic season of podcasting.
We are. I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.